So let's talk about cellular transport mechanisms. Now, what we're looking at, membrane transport, is the ability to move substances back and forth into and out of a cell. This is important because the cell has to have certain reactions that occur, certain activities that can occur inside the cell, and those reactions don't occur if you don't have the right stuff there. So we have means of getting stuff in and out of cells that optimize the function of the cell. We've got two basic methods of transport. We have passive transport, which doesn't require any energy, and that's great. Anything you can get for free is much better than having to pay for it. And we use these transport mechanisms actually on a couple of processes that are very important, like getting oxygen into cells, getting carbon dioxide out of cells, movements of some small substances. So they don't require energy. We have osmosis, which is specifically the movement of water. So when I'm talking about the movement of water in any test question, anything like that, we're talking about osmosis. We have diffusion, which is the movement of solutes. If water is a solvent, then we've got stuff that's dissolved in it that are solutes, like sodium, potassium, oxygen, carbon dioxide, glucose, all sorts of stuff that are located in the plasma and in the interstitial fluid that surrounds cells. So we have diffusion of these solutes. And then we have facilitated diffusion. And in facilitated diffusion, you have to have a doorway, basically something the appropriate size and shape to allow substances in, but it still doesn't require any energy from the cell. And then we have filtrations. And in a filtration, what you're looking at is hydrostatic pressure. That is, well, if you make coffee, the force of water as gravity pulls it downward and through the filter, or through the filter. Now, active transport requires ATP. That is the energy currency of the cell. Uh, Now, let's take a look at a little terminology here. Solutions and transport. A solution is a mixture of a couple of components and a fluid component. So we've got blood plasma and we've got water in the plasma, which is the solvent. And then we have the solutes. And as I said before, these are sugars, these are hormones, these are gases, but they're all stuff that's dissolved into the fluid. Now we have a couple of different fluid components within our body. We have intracellular fluid, which is the stuff that's actually inside the cells, like nucleoplasm, cytosol. And then we have interstitial fluid, which is stuff outside of the cell. And all of our cells are bathed in fluid. What we've got here, interstitial fluid on the exterior of the cell. There's also extracellular, an extracellular component that's located in blood plasma. We don't really consider that the interstitial fluid. We'll talk about that a little bit later on. Now, plasma membrane, that is the phospholipid bilayer that we have that makes up our cells, is selectively permeable. That is, it can pick and choose what comes in and out. Sometimes it does this just by opening the appropriate door. Sometimes it actually actively pulls stuff in. And then there is this semi-permeable characteristic, semi-permeable being slightly different than selective permeable. That is, semi-permeable will allow stuff through that is small enough to pass through gaps. So a good example of something that's semi-permeable is dialysis tubing, because it has small openings just big enough to allow toxins to exit. Now, this permeability does affect the movement in and out of the cell, so we can end up with a vastly different environment inside the cell than outside the cell. That's important as well. We have to have a environment set up as I said before, to optimize the functioning of the cell. And if it was just the general mishmash of stuff that's outside of the cells, we wouldn't really have very much of a function. So, passive transport processes. The first one we'll talk about is diffusion. And with diffusion, particles tend to distribute themselves over time so that we get to a point we call equilibrium. That is, everything's balanced out. So we can have a high concentration of a substance, but eventually it will spread out. And at that point, we have what we call an isotonic solution. A hypertonic solution is where we have a concentration heavier in one area than another. And a hypotonic solution is where we have a concentration less in one area than another. Now, what we have is a tendency for things to move from an area of high concentration to a low concentration. And this is all a product of random movement of atoms and molecules. Now, every atom, every molecule has a certain amount of energy. And that energy was imparted to it, call it the Big Bang, call it whatever. But the fact is that we have this energy that manifests itself as slight movement. Everything kind of vibrates just a little bit. So we have atoms that are just shifting back and forth randomly. And when they do this, they may bump into other atoms, and they can impart energy to them or take energy away from them. But in general, they're just randomly moving and spreading out slowly. So this is what we call moving from an area of high concentration to a low concentration, or down a concentration gradient. Now, how this works is, as I said, we've got this random movement of particles everywhere, and this movement is called Brownian motion. And I've got a little graphic here that will show you how this works. And here you can see these are nanoparticles, very small particles that are moving randomly around. But so there are different things that can modify how things diffuse. If we have a high concentration of stuff in one area and a low concentration in another, depending on how high the concentration is, the faster it's going to spread. I think a lot in terms of pool tables and things like that. So if I have a rack full of balls, they are highly concentrated. Now, if I hit that rack of balls, they're going to spread out randomly and fairly quickly. On the other hand, if I'm shooting at single balls, then I'm not going to get as much of a movement. So concentration is one factor. As a different example, I have an aunt who has pretty much no sense of smell whatsoever. And she tends to just soak herself with Chanel number 5, which the fact is that she loads herself up with this, and because she's so covered with it, it spreads very rapidly. I remember being in church with her, and she would be 5 rows away, and I could smell her. If she had not put that much on, if she'd put the amount of perfume on that a normal person would have, you might have gotten a whiff of it if you got close to her. So concentration does impact how much something, or how quickly something will spread. Distance traveled is another factor. Let's go back to our pool table. If I queue up and I hit those balls, at first there may not be any, well, there won't be any balls at the end of the table that I'm at. But over time, those balls spread out, and I will eventually get some up toward the far end. But initially, when I queue up, they're all kind of in the area where the rack was before they spread out. Temperature is a factor. And when I said that we have movement, Brownian movement, well, Brownian movement is a reflection of the energy that's in things, and energy and temperature are more or less the same. So I add energy, I add temperature. And the higher the temperature, the faster things are going to spread. Now, in our bodies, we can't alter the temperature, so we can't make reactions happen faster by raising our temperature or lowering our temperature significantly. So we have other mechanisms that we get into when we start talking about proteins and enzymes. The fact is that our bodies are optimized to a certain temperature so that they work the best. So temperature is a factor. In general, what you'll see is that if you raise the temperature, you're going to increase the Brownian motion, and you're ultimately going to have things spread faster. Now, size of particle is another factor. Going back to my pool table analogy, if I am imparting energy by queuing off and hitting the rack, what's going to happen is I've imparted more or less the same amount of energy to all the balls on that table. And they're all going to move randomly at more or less the same rate. Now, if I took that same rack of balls and in the middle of it I stuck a bowling ball, well, I can queue up and I can hit those balls, and the pool balls are going to move around at a certain rate. But that bowling ball is not going to move nearly as fast. And I've imparted the same amount of energy to that bowling ball as I have the other balls, but because of its size, it just doesn't move as quickly. There are a couple of factors there. First off, the amount of energy absorbed for the mass that's going to, the higher the mass, the less movement you're going to see. And when you have large particles, they actually have to shove stuff out of the way as they move around. So let's take a look at these different types of diffusion. We've got simple diffusion, and simple diffusion doesn't require any kind of special opening. It doesn't require anything. It just happens. It is an unassisted process, and things that are dissolved in a material can pass through small pores that are in the cell membrane, for instance. And these just pass up and down their concentration gradient, or down their concentration gradient. Now, things that can do this, lipid-soluble materials can go through the phospholipid bilayer. So a substance like alcohol, which is lipid-soluble, passes directly from the bloodstream right into the cell without any control or regulation whatsoever. This is as much as we can cover in this video. We will continue on this topic in subsequent videos. Thank you.